Yes, ma'am. It's now it's fine, ma'am. Dr. Bhuneshwari, ma'am. Now it's fine.
Yes,
Started
ma'am.
recording.
Yes, ma'am.
A very good afternoon to all. Myself, Dr. Amsaveni, working as Assistant Professor in the Department of Biotechnology, Kongunadu Arts and Science College, Coimbatore. On behalf of PG and Research Department of Biotechnology and on my own behalf, I feel happy to welcome you all for this International Alumni Webinar on polyphenol-based surface engineered nanomaterials for improved therapeutics. With the blessings of our beloved President Dr. M. Archami sir and the unconditional support and motivation of our guiding star Dr. C.A. Vasugi ma'am, Secretary and Director, Kongunadu Arts and Science College, we have organized this webinar for the benefit of students as well as for the research community. Initially, we have organized an alumni webinar series during the first week of June 2020, and the outcome was like the students, scholars, and the faculty members, they got a chance to interact with the resource persons, and the resource persons, they also accepted for the collaborative research in future. Thank you so much, ma'am, for the opportunity. With this note, may I now invite Dr. V. Bhuneshwari ma'am, Associate Professor and Head, UG Department of Biotechnology, and one of the organizing secretary of this webinar to offer the welcome address. Ma'am, please.
A very good afternoon to all. As one of the alumnus of this prestigious institution, I feel proud to welcome you all on behalf of the management, Department of Biotechnology, and my own behalf for this one-day international alumni webinar on polyphenol-based surface engineered nanomaterials for improved therapeutics, organized by Department of Biotechnology today, 13/6/2020. First of all, I dedicate my welcome in absentia to our beloved President, Dr. M. Archami sir, for his immense support and encouragement rendered in the growth of this department. I warmly welcome the guiding star of our beloved Secretary and Director, Dr. C.A. Vasugi ma'am, for her idea and motivation to organize this alumni webinar and providing a platform to share our views. I welcome you, ma'am. I welcome dedicated and sincere principal, Dr. M. Lakshmanaswamy sir. I gladly welcome our distinguished and enthusiastic Chief Executive Officer, Unneeded Course, Dr. B. Chinnusamy sir. I welcome you, sir. I express my delighted welcome to our respected and motivating Dean R&D, Dr. S. Paulsamy sir, and supporting Dean Academics cum HOD, Biotechnology PG, Dr. S.R. Madan Shankar sir, for their constant support and immense help rendered in all our endeavors. I cordially welcome our guest of honor, our prestigious alumnus, Dr. G Sathish Kumar sir, postdoctoral researcher in School of Materials and Energy, Southwest University, China. I welcome you, sir. He has readily accepted our invitation in spite of his busy schedule for this webinar. I kindly welcome all the participants for their interest to this webinar. I warmly welcome the organizing secretary, Dr. R. Amsaveni, Assistant Professor in Biotechnology, session coordinators, participants from different institutions in India and as well as in abroad. Once again, I welcome you all. Now I invite the organizing secretary of this webinar, Dr. R. Amsaveni, Assistant Professor in Biotechnology, to introduce our guest of honor. Ma'am, please.
Thank you, ma'am. Thank you for your warm welcome. I take this opportunity to introduce today's resource person, as well as my MSc Biotechnology classmate at Kongunadu Arts and Science College during 2008 to 2010, the most eminent person, Dr. G Sathish Kumar. Now he's a postdoctoral researcher, School of Materials and Energy, Southwest University, China. He did his BSc Biotechnology at Sengundar Arts and Science College, Tiruchengur, MSc Biotechnology at Kongunadu Arts and Science College, Coimbatore, MPhil Biotechnology at Bharathidasan University during 2010 and 2011, PhD in Biotechnology at Bharathidasan University during 2012 to 2018. He was a research fellow at Bharatdasan University during 2017 to 2018 under DST Promotion of University Research and Scientific Excellence Program. His major research contributions and awards include he developed different biogenic nanomaterials and nanocomposites for various biomedical applications. He also received DST ITS Travel Grant Award. His technical and specialized skills include interpretation skills for characterization of materials using XRD, SEM, TEM, UV-VIS, and FTIR, and also handling instruments like a few to mention FE, SEM, HR, TEM, and XRD. He has attended various conferences, seminars, and workshops, and he has published his research in various international reputed journals. He has a cumulative impact factor of around 86.5, and his total citations is around 1020. We are very happy to have you with us, sir, for this webinar. And before we start the session, now may I request our most beloved and ever guiding personality, Dr. V. Chinnusamy, sir, Chief Executive Officer, Kongunadu Arts and Science College, for the inaugural address. Sir, please. Sir, I request.
We are very good afternoon to one and all. We are very happy that the Department of Biotechnology is organizing so many seminars, especially in this COVID-19 session, that is webinar series. Dr. Bhuvaneswari HOD of Biotechnology are not only organizing webinars in biotechnology, Institution Innovation Cell, she is organized so many seminars, and Amsavani is a very great specialist in biotechnology. Her classmate is now on the line. We are very happy that we are handling so many hands-on training programs. Every year we are giving so many hands-on programs on PCR, real-time PCR. Now people are speaking about real-time PCR. Many of the people in our college and other colleges from various universities, they attended our hands-on training programs and benefited very much. During this COVID-19 session, these ideas and this technology is highly useful to all to know about what is COVID-19 and how to prevent it, how to identify it. This type of seminar is highly useful for us. For this, our today's chief guest is here from China. He is addressing our students as well as professors about this program. Okay, thank you very much for organizing such a wonderful program. We wish this program a grand success. Thank you, one and all.
Thank you so much, sir, for your nice words and gracing the occasion. Thank you so much. Ma'am, shall we start the session, ma'am?
Yes, ma'am. Yes, ma'am. You can invite the guest of honor.
Okay.
To start the session.
Yes, ma'am. Now I invite our resource person, Dr. G Sathish Kumar, sir, to take over the session on the title Polyphenol-based surface engineered nanomaterials for therapeutics. Sir, please.
Thank you very much for the kind introduction. Very good afternoon to everyone. So I'll share my slides.
Dr. Bhuvaneswari, ma'am.
Yes, ma'am.
Ma'am, you just close that brochure, ma'am.
Okay.
Very good afternoon to everyone. First of all, I would like to thank our respected President, Dr. Orichami, for and other members of the organizing committee for giving me this wonderful opportunity to present my research in front of this wonderful audience. Uh, so before So my research is focused on polyphenol-based surface engineered nanomaterials for improved therapeutics. So before going to the main research, let me brief some introduction about nanotechnology. So as we know that nanotechnology involves with design, synthesis, and manipulation of atoms or molecules with the size of 10 to the power of minus 9 meter. So any material which is under 1 to 100 nanometer is called as a nanomaterial. So the word nano is derived from the Greek word nanos, which means small. And the term nanotechnology was coined by the Japanese scientist Noria Taniguchi in 1974. So why we need to go for nanomaterials? So as we know that silver in any form will give antibacterial property. So silver nitrate will give antibacterial property, and bulk silver will also give antibacterial property. So why we need to go for a nanomaterial? Because nanomaterial will give a unique, will be having a unique size, safe, and surface properties. These properties can be used to load more amount of drug, and these unique properties will also give improved biological activities. Like, for example, take an aspirin drug and put it in water. It will take at least 2 minutes to dissolve in the water. The same aspirin drug, if you grind it and make it into powder and mix it with water, so it will get hardly 1 minute to dissolve. So that is the positive thing about the nanomaterials. So the nanomaterials will be having a large surface area to volume ratio. This large surface area to volume ratio will allow us to load more quantity of drugs. And also these nanomaterials will be having unique optical, electrical, mechanical, biological properties than the macro scale counterparts. So nano is not a new thing. It is already exist in nature, as well as there are some ancient sculptures having this nano technology. So here, the emittance of colors in the butterfly and peacock feathers are due to the scattering of light by the existing nanostructures. These nanostructures not only makes it beautiful, but also it gives a protection from the dust particles. So the gold nanostructure in the Chinese painting emits different colors. So, and another example is the Lycurgus cup. So when an instant light passes through this cup, it will give red color. It will emit red color. Whereas in the absence of light, it will give green color. So nano in medicine. In our Indian traditional medicine, we are using basmas. These are all nothing but the nano formulations. So the very well-known example is the Swarna Basma, that is the gold nano formulation. So the synthesis of nanomaterials, broadly categorized into two types. One is top-down approach and another one is bottom-up approach. In the top-down approach, the bulk material will be sliced into nanoparticles, whereas in the bottom-up approach, the atoms will be assembled to form a nanoparticle. The process is called a nucleation process. So these nanomaterial synthesis is further can be achieved through different methods, physical methods, biological and chemical methods. In physical, there are different methods are there: vapor deposition, microwave irradiation, ablation, sonochemical method, and irradiation methods. And chemical methods is also used for various nanomaterial preparation. There are some chemical methods or chemical reduction using sodium borohydrate and hydrogen hydride. And these physical and chemical methods are largely limited with the use of high energy consumption, and the use of toxic chemicals will give adverse effect to the environment. So in order to avoid these limitations, nowadays people were trying for the biological mediator synthesis of nanomaterials. So in biological mediator nanomaterials, there are different biological entities like bacteria, fungi, yeast, actinomycetes were explored for the synthesis of various nanomaterials, metal oxides, and 2D nanomaterials. So among the biological nanomaterial synthesis, plant-mediated synthesis was gaining much attention because of its simplistic process. And if we take in microbial mediated synthesis, we need to go for microbial culture maintenance, and it also requires tedious downstream processing. So people are fascinating towards the plant-mediated synthesis of nanomaterials. These plant-mediated nanomaterials can be achieved with a different size, shape, and topography. And these materials were largely used in different biomedical applications like drug delivery, theranostic devices, hemo and photothermal therapy, antimicrobials, antioxidants, tissue engineering, regenerative medicine, and also for the diagnostic applications. So whenever, if you want to synthesize a nanomaterial, you should think about the two parameters. One is the reducing agent and another one is the capping agent. Here, the reducing agent will play a role in the reduction of the material, the reduction of the bulk material into nanoform. And whereas the capping agent will be covered on the surface of the nanoparticle, will provide stability to the nanoparticles. There are plenty of medicinally important plant entities have been explored for the synthesis of various inorganic nanomaterials. And all those reports mainly suggest that the plant active polyphenols and other reducing sugars and proteins, they were playing a major role in the reduction and as well as the stabilization of the nanoparticles. So these interaction of these active biomolecules with the nanoparticle will also provide improved end product, which means these plant extracts, if you test these plant extracts for any biological applications, as compared to the plant extract, the nano combined, the plant extract that has been in the nano formulations is giving more biological properties. So with this objective, we have initially we have screened plant Morinda citrifolia, commonly known as the noni plant, for the synthesis of silver nanoparticles. So in this study, we have used the leaf extract of this particular plant. The leaf extract is prepared through the simple decoction method. That is the specialty of the plant-mediated nanomaterials. You don't need to go for a large extraction process or a tedious extraction process. Simple decoction method is enough to synthesize the to extract the plant leaf components. So the extracted leaf components were used with silver nitrate. We have used the silver nitrate as the precursor and obtained a nanoparticle with the size range between 10 to 60 nanometer. Here you can see the polydispersed nanoparticles, and we can see the particles are quite aggregated. Because in plant-mediated synthesis, we cannot predict the exact active compound, which plays a crucial role in the synthesis and reduction and stabilization. Because the plant extract, obviously, it will be having a mixture of compounds. So we cannot say this particular compound is involved in the reduction and stabilization process. And also in plant-mediated synthesis, we cannot predict the mechanism of reduction. So, nanotechnology, basically, it is an interdisciplinary research. So we have to compromise. We have to, get all the ideas in all disciplines. Like we have to get the physical information about the material and the chemistry of the material, and as well as the biological properties of the material. So here in plant-mediated synthesis, it is very tough to predict the exact mechanism of synthesis. So we have initially we have started with plant-mediated synthesis. We have screened a lot of plants, medicinally important plants, and also some of the commercial plants for the synthesis of different nanomaterials. We have synthesized different metal oxides and also 2D nanomaterials. Recently, we are exploring on the 2D nanomaterials, biogenic 2D nanomaterials. So in another study, we have used Dendrophthia falcata plant extract. This is a neem a parasitic plant. This plant grows on the neem plant. So it ultimately it will kill the neem plant. So we thought we can use this plant as a potent bioresource to synthesis silver nanoparticles again. But this time we would like to explore for anti-cancerous property of the nanoparticle. So here we found that as compared to the crude plant extract, the silver with the plant extract is giving more anti-cancer activity against the breast cancer cell line. So here also you can see the particles are largely aggregated. It is due to the, in synthesis, biogenic synthesis, not only the capping agent or the reducing agent plays a role. There are certain physiological conditions, the reaction parameters, that is also plays a major role in the size and the shape of the nanomaterials. So here we found that the synthesized silver nanoparticles were in the size range between 5 to 45 nanometer. So in another study, we explored the fruit extract. This time we have explored the fruit extract of the plant Coropita guineensis, and this time we have synthesized gold nanoparticles. And here you can see the gold nanoparticles with a mixed shape and size. It has both triangle and spherical sized particles with the size range of 25 plus or minus 6 nanometer. So here also we cannot say this is mono-dispersed particles. In another study, we have explored the same fruit extract for the palladium nanoparticle synthesis. Here, the HRTM micrograph clearly shows that the particles are polydispersed in nature, and here we have used palladium chloride as the precursor. And moreover, in this study also, we cannot predict the exact biocompound which is responsible for the synthesis and stabilization of the nanoparticles. And here we have tested this palladium nanoparticle against human lung cancer cell line. Here also, it was very clearly evident that the plant as compared to the plant extract, the synthesis nanoparticles is having more antibacterial as well as anti-cancerous properties. So based on our preliminary screening with different plant extracts and the plant extract that has been reported by different research groups worldwide, it was clearly evident that most of the, almost 80% of the plant-mediated synthesis in plant-mediated synthesis, the polyphenols plays a major role in the reduction and stabilization of the nanoparticles. By considering this fact, we thought instead of going for a crude plant extract, why can't we try a single polyphenol compound for the synthesis and stabilization of the nanoparticles? So as we know that there are plenty of active metabolites have been isolated, identified, and reported as a potential drug candidature for the different disease, right from cancer and the various potent scourges of the mankind, like cancer, tuberculosis, and other bacterial infections. So even though these active metabolites have been were reported as a potential drug candidature. There are certain limitations are there for which hinders the clinical translation of these polyphenols or the active metabolites in the successful clinical translation. The translational medicine, in translational medicine, these polyphenols cannot be successful because of its limitations like poor solubility, bioavailability, and requirement of a high amount of dosage. These limitations hinders the success rate of the the polyphenols in clinical translation. So we thought the combination of the nanoparticle with the polyphenol will concurrently will improve the polyphenol as well as the synthesis of nanoparticles. So we have combined these two objectives together, and we know that there are, as I mentioned, there are different classes of polyphenols, especially the phenolic acids and flavonoids, were explored for different biological applications like antibacterial, antiviral, anti-tumor, antioxidant, anti-inflammatory, anti-allergic, and anti-diabetic as well. So this, with this objective, there are several polyphenol-based nanomaterials have been already reported over the past decade. The wide range of polyphenol-based materials, especially polyphenol-based colloidal silver and gold nanomaterials, and polyphenol encapsulated polymeric nanomaterials, and polyphenol functionalized 2D nanomaterials, and other surface coatings were developed for different types of biological applications, right from the treatment of various diseases and for diagnosis of various diseases, and particularly for the cancer therapy. So these polymer-based nanomaterials have been developed for different applications like drug delivery and regenerative medicine, and also for photothermal therapy and photodynamic therapy, gene therapy, chemotherapy. So with this objective, we have selected flavone known as the chrysin. This particular flavone is having excellent anti-cancer activity. So this is almost this particular flavone has been tested with almost all type of cancer cells, and it is under clinical trial. Even though it has been known that it was known that this particular chrysin can inhibit the growth of cancer cells through apoptosis, cell cycle arrest, and inhibition of the angiogenesis and the invasion of the metastasis as well. There are certain limitations which limits its application in the clinical trial. Those are high molecular size, poor absorption, loss of stability, bioavailability, and efficacy. So in order to improve the qualities of chrysin, we thought we can incorporate or functionalize this chrysin with nano formulations, and also we can use this chrysin because this chrysin is having a hydroxyl group, which can reduce the metal ions into metal nanoparticle form. So with this objective, we have selected this chrysin to reduce silver and gold nanometals, metal ions to metal nanoform. As I mentioned earlier, in the synthesis of metal nanoparticles, not only the reducing agent and stabilizing agent plays a role. Here we have optimized all the parameters like the quantity of reducing agent, the quantity of precursor, that is silver nitrate or gold chloride, whatever, based on the nanomaterial. Also the pH of the reaction medium. And another important parameter is the stoichiometric proportion of the reaction mixture, and the temperature and the incubation time, that is the reaction time. These parameters largely influence the size and the shape of the nanomaterials. So instead of whenever we synthesize nanomaterials, particularly these optical nanoparticles. We need not to go for high-end experiments like HR10, which is a costly experiment. So we can predict the size of the nanoparticles based on the color formation as well as the surface plasmon resonance, which is known as the SPR effect. This SPR effect is due to the collective oscillation of the electrons on the surface of the nanoparticles. So in this UV visible spectrum, here the broad spectrum clearly indicates the polydispersity of the nanoparticles, whereas the intense peak clearly indicates the presence of monodispersed nanoparticles, which means the particles are in a narrow size range. For example, 5 to 10 nanometer, 5 to 15 nanometer. Whereas the broad spectrum clearly indicates that the particles are polydispersed in nature, which means the particles are in a 10 to 60 nanometer, 10 to 80 nanometer. So based on this SPR peak, we can predict the size and shape of the nanoparticle, as well as we can optimize all the reaction conditions like pH, temperature, and the incubation time also. Similarly, based on the color also, we can predict the size of the nanoparticle. Here, the gold nanoparticle with red color indicates that the particles are in the range of 3 to 30 nanometer, and the violet color indicates that particles are above 100 nanometer. So based on that, we can predict the size, and we can control the size of the particles. Not only for this particular reducing agent. So if you are using a plant extract also, we have to fix all these optimum conditions. Then only we can able to get better quality end products. So yeah, as I mentioned earlier, the size and the shape of the particles largely influence the biological fate of the nanoparticle. So we have to be very careful, and we have to give more concern to the size and the shape of the particle. Apart from the size and shape, another important criteria is the surface chemistry. The surface chemistry and the surface properties of the nanoparticles can be studied through different spectroscopic analysis. The preliminary spectroscopic analysis is usually will go for FTR analysis. So this will give some important information about the functional group which is responsible as well as which is involved in the reduction of metal ions. Here, as compared to the free chrysene, that is the chrysene molecule, the chrysene that is being functionalized with the nanoparticle shows the involvement of hydroxyl and carbonyl group. So an electron from this hydroxyl group, the hydroxyl group of the chrysene donates an electron to the Ag plus or Au3 plus ions, which convert it into Ag0 or Au0, and the carbonyl group will be in a coordinated linkage with the nanoparticle. So the quinone form of the chrysene is being functionalized on the surface of the nanoparticles. So this is the important thing about the polyphenol based nanoparticle. At least we can able to predict the possible reduction mechanism involved in the reduction and stabilization of the particles. Whereas in plant mediated synthesis, we cannot predict even the possible reduction mechanism. So this is the FTR is a preliminary study. So we have to go for further analysis like X-ray photoelectron spectroscopy. So this X-ray photoelectron spectroscopy will give information about the elemental composition of the nanoparticle. So here the core level spectrum shows the binding energy at around 368 and 374 electron volt, which clearly shows the presence of silver as a major element in this combination, in this formulation. And the other, the C1C carbon spectrum, the deconvoluted carbon spectrum shows the presence of different functional groups. These functional groups are from the chrysin. So the core level spectrum were located at around 284, 285, which shows that the presence of the functional groups. And also this confirms the successful functionalization of chrysin onto the surface of the nanoparticles. Similarly, for gold nanoparticle also, we have observed the core level spectrum at around 86.7 and 83.3, which clearly shows the presence of gold as a major element, and also the other functional groups clearly indicates that the chrysin is being functionalized on the surface of the particles. This is a very important analysis. Size, we have studied the size, safe, and also the surface chemistry. Another important parameter is the stability. So whenever we develop a nanoparticle, it should be stable for a long period of time without any aggregation. So here the large negative zeta value clearly indicates that the particles are highly stable in aqueous medium. So because of the large negative charge on the surface, the particles will repel each other. So it will prevent the aggregation of the particles. So if two particles coming close together, so because of the negative surface charge on both the nanoparticle, it will repel each other. So it is very important to synthesize particles with either large negative zeta value or positive zeta value. So here we found that the synthesis silver and gold nanoparticles were having large negative zeta value. And another study is the dynamic light scattering, which gives some information about the hydrodynamic diameter, which means the diameter of the particle the size of the particle which is in the aqueous medium. XRD. Coming to XRD, XRD is used for the analysis of crystalline nature of the nanoparticle. So this study will give information about the crystallinity and the crystalline phase of the nanoparticle. So this XRD will be the X-ray diffraction planes will be interpreted with JCPDS standard JCPDS value. So here we found that the diffraction value corresponds to the phase-centered cubic silver as well as the phase-centered cubic gold nanoparticle. So this is also another important thing that we have without any mixed phase of hexagonal or other mixed phase. We have identified a single phase nanoparticle. This is because of using the polyphenols, the single biocompound as a reducing agent. The size and the topography of the nanoparticle were further analyzed with the AFM image, AFM analysis. So this is AFM and both AFM and SEM analysis are the preliminary screening of the size of nanoparticles. But the high resolution transmission electron microscope will give the exact size range of the nanoparticle. So for this analysis, we will coat the nanoparticle on the copper grid, and then we'll perform the TEM analysis. Here we found that the synthesized gold nanoparticles are largely monodispersed in nature. And here you can see the high resolution image clearly shows the lattice fringes, which corresponds to the face-centered cubic crystalline nature of the particle. And also the selected area electron refraction pattern also shows the presence of face-centered cubic crystalline nanoparticles. This is a supportive data for the XRD analysis. So the particle size distribution shows that particles are in narrow range with a size range of around 5 to 25 nanometer, with an average size of around 14 plus or minus 6 nanometer. And the EDAX analysis, this is EDAX analysis. This is a preliminary screening of the elemental composition. Earlier we saw the XPS analysis. That is the there you can find the exact elemental composition, but this EDAX is a preliminary screening of the elemental composition. Here we found the maximum signal for silver, and the other carbon and oxygen are due to the presence of chrysin, and the copper is due to the grid that we have used for the analysis, TEM analysis. Similarly, the gold nanoparticles are also in monodispersed distribution, and we found that the gold nanoparticles are in both spherical as well as in some ovoid shape, and the high resolution images clearly show the lattice fringes corresponds to the face-centered cubic crystalline gold nanoparticles, and the particle size distribution shows the presence of particles with the size range in between 5 to 20 nanometer, with an average size around 6 plus or minus 2 nanometer. And the elemental composition also, the EDAX analysis shows the presence of gold, carbon and oxygen. The carbon and oxygen are due to the presence of chrysin. So here you can compare both the plant mediated nanomaterial as well as the nanomaterials that have been synthesized using the polyphenol. So as in my earlier slide, I have shown 3-4 plant mediated nanomaterials, and those nanomaterials are largely aggregated, but here you can see individual nanoparticle without any aggregation. Here you can see the overlapping of the particles, but these are due to the overlapping, not the particles are aggregated. We cannot say these particles are aggregated. The particles are in highly stable without any aggregation. So another interesting study by Dr. Satish Kumar et al., who is our collaborator, and they have used a density functional theory based, which means they have used in silico analysis to identify the reduction mechanism, flavonoid mediated metal nano reduction mechanism. So they found that the 5' hydroxyl group of the myricetin large involved in the reduction, as well as the carbonyl group of the myricetin interacts with the gold ions and involved in the functionalization process. So we have experimentally we have proved the functionalization process, but here they have used density functional theory. These results are highly corroborate with our results that I have shown in the FTR analysis, as well as the possible reduction mechanism that we have predicted, which is confirmed with this study. So we have successfully synthesized the metal nanoparticles, as well as because of the time limit, I couldn't incorporate the metal oxides and their nanomaterials. But interestingly, we have synthesized polyphenol-based 2D nanomaterials. The 2D nanomaterials are the recent trend nanomaterials. Different types of 2D nanomaterials like graphene, M-genes, black phosphorus, and transition metal dialkogenides. And these are all the recent nanomaterials that have been widely explored for different biomedical applications. So with the success in metal nanoparticles, we have explored the nanobiotechnological potential of this particular flavonoid chrysin for the synthesis of 2D nanomaterials. It is not like metal ion reduction and forming a metal nanoparticle. 2D nanomaterials, we have to go for both oxidation as well as the reduction process. It involves with the both oxidation and reduction process. So this is the scheme shows the synthesis of 2D nanomaterials through the sustainable green chemistry approach. And here, for the synthesis of graphene oxide, so we have to prepare the graphene, the synthesis of reduced graphene oxide. We have to prepare the graphene oxide from the pristine graphite, which means the raw graphite materials, the raw graphite flakes will be oxidized to graphene oxide. Then the graphene oxide will be used as the precursor for the synthesis of reduced graphene oxide. And here we will use chrysin as a reducing agent to reduce graphene oxide into reduced graphene oxide. Here you can see the multi-layer graphite flakes will be oxidized to graphene oxide, and from the multi-layer graphene oxide, monolayer reduced graphene oxide will be synthesized using our chrysin as a reducing agent. And after successfully synthesizing the reduced graphene oxide also, we have got an idea to synthesis both graphene oxide as a reduced graphene oxide as well as the metal ion in a composite formation. So here we have used both graphene oxide and silver nitrate for the synthesis of silver nanoparticle decorated graphene oxide, as well as for the gold, we have used chlororic acid and graphene oxide to synthesis gold nanoparticle decorated reduced graphene oxide nanocomposites. In both the cases, we have used chrysin, and you can see, and the chrysin is capped on the nanoparticle surface. So this is the synthesis protocol. So as I mentioned earlier, we cannot synthesis the reduced graphene oxide or its composites directly from the pristine graphite. So we have to go for a oxidation process. The process is called modified Hammer's method. So in this method, we will be using sulfuric acid and potassium permanganate as oxidizing agent to convert pristine graphite into graphene oxide form. So from the graphene oxide, all the nanomaterials and nanocomposites will be synthesized using chrysin. So as I mentioned earlier, we have to fix all the reaction conditions like pH, temperature, the reaction time, and the quantity of the graphene oxide, as well as the reducing agent chrysin and the stoichiometric proportion. All these parameters have to be optimized. Then only we can able to get precise nanomaterials and nanocomposites. And after the synthesis, obviously, we have to go for all the instrumentation, starting from UV visible and all the instrumentation that I have mentioned earlier. The same analysis have to be performed to confirm the synthesis of all nanomaterials and nanocomposites. So in graphene oxide synthesis, it is not like a silver or gold nanoparticle where you can easily predict with with the UV visible spectroscopy. Here, as compared to the graphene oxide, the peaks at around the absorption around 234 and 314 were converted into 266 nanometer. This is due to the restoration of the carbon-carbon backbone. For the clear understanding, I will explain more clearly. So in the pristine graphite, we will be having the carbon-carbon backbone. So after the oxidation, so the sum of the oxygen functional groups will present in the graphene oxide. So these two absorption peak indicates that presence of the oxygen functional groups due to the oxidation process. And again, after the reduction, the carbon-carbon backbone has to be restored, which means the pristine graphite form, the multi-stack graphite will be converted into monolayer graphite through the oxidation process and the reduction process. So both the graphite and the reduced graphite will be having the similar composition, but here the graphite is multi-layer, but here this is a monolayer. And in case of the nanocomposites, so we have used silver nitrate and gold chloroauric acid for the synthesis of silver nanoparticle decorated nanosheets, graphene nanosheets, as well as the gold nanoparticle decorated. Decorated means the hybridization process. The hybridization is broadly categorized into two types. One is a ex situ hybridization and another one is in situ hybridization. I don't want to discuss more elaborately about the hybridization process and these things because it will be confusing. So here we have clearly confirmed that the presence of gold nanoparticles through the SPR phenomena that I have explained in my previous slide. The peak located around 430 nanometer clearly indicates the presence of silver nanoparticle, and here the peak around 560 nanometer clearly indicates that the presence of gold nanoparticle on the surface of the nanosheets. So again, we have studied the stability of the nanoparticle, and here you can see as compared to the silver and gold nanoparticle, individual silver and gold nanoparticle, the reduced graphene oxide is having large negative zeta value, which clearly indicates that the nanosheets are highly stable. And for the silver and gold nanoparticle also, with the presence of these reduced graphene oxide nanosheets, the silver and gold nanoparticles surface charge is also increased, which means that as compared to the individual silver and gold nanoparticles, the composite, the nanoparticles are in the composite. The nanoparticles that are decorated on the surface of the nanosheets, reduced graphene oxide nanosheets, are more stable than the individual nanoparticles. So here also, as we have used single flavon, we can able to predict the mechanism of reduction that is the conversion of graphene oxide into reduced graphene oxide. So as I mentioned earlier, the graphene oxide will be having the oxygen functional groups like epoxide and hydroxyl functional groups. So here then chrysene will act as a nucleophiles, and through the epoxide nucleophilic ring opening mechanism, the oxygen functional groups will form an intermediate, and then the chrysene functionalized reduced graphene oxide will be formed. This kind of prediction of the possible mechanism can be only achieved in the polyphenol based or a single biocompound based. Not only polyphenols, we can also explore different reducing sugars, carbohydrate polymers also for the synthesis of nanoparticles. As we were using polyphenols, I'm mentioning more about the polyphenols. Those who are interested in this kind of research, they can explore different other biocompounds like carbohydrate polymers or other active metabolites, even proteins. So this is the FTR spectrum of the nanocomposites. Here also, in all the nanomaterial and nanocomposite preparation, we have clearly found that the hydroxyl and the carbonyl functional groups of the chrysene largely influence the synthesis of the reduction as well as the functionalization of the nanomaterial as well as the nanocomposites. Now here we will get a question. As I mentioned earlier, the chrysene is having excellent anti-cancer activity. What about this form of chrysene, which means the chrysene is being functionalized on the surface of the nanoparticle? Will it have the same kind of anti-cancer or antibacterial application? The answer is yes. The quinone form of the chrysene will also give the same kind of antibacterial and anti-cancer applications. Moreover, here the nanomaterial and the chrysene will give a synergistic effect, and which will give more bioactivities as compared to the free chrysene. So this is an interesting study, the Raman spectroscopic study. So the experimentation, the instrumentation is depends on your material. This Raman spectroscopy, you need to go for Raman spectroscopy if you are synthesizing a metal or metal oxide nanoparticle. It depends on your research, but for the 2D nanomaterials, the now Raman spectroscopy is a mandatory study. So here we can able to see based on their intensities. So the increased intensity clearly indicates the conversion of multi-layer graphite into single layer graphene oxide, reduced graphene oxide. And the 2D band clearly indicates the disappearance of 2D band clearly indicates the conversion of graphite into reduced graphene oxide. Similarly, for the metal decorated nanosheets, nanocomposites, the increased intensity due to the optical property of the silver and gold nanoparticles, and also because of the surface plasma resonance effect, it shows increased intensity. So again, we have to analyze the elemental composition and the complete composition of the material. So we have to perform the XPS analysis, which means the X-ray photoelectron spectroscopy, to get an idea about the complete formulation and the composition. Here you can see the carbon core level spectrum clearly shows the restoration of carbon-carbon band, and it also conveys that the graphene oxide is successfully converted into graphene, reduced graphene oxide, and the presence of all these functional groups uh, uh, highlights that the chrysene is being functionalized on the surface of the nanosheets. In case of the composites, uh, here we can see the core level spectrum of silver indicates the presence of silver that is being hybridized on the surface of the nanosheets, and all the functional groups due to the presence of chrysene. Similarly, for the gold nanoparticle also, the core level spectrum clearly shows the presence of gold as a major element, and other functional groups indicates that the presence of chrysene. Here also XRD analysis give some information about the conversion of pristine graphite into monolayer reduced graphene oxide. And also here we can, in composite preparation, based on the diffraction planes of, here we cannot see any diffraction planes because the graphite is, it is not a crystalline material. But after the hybridization of the crystalline material like silver and gold, we can see the diffraction planes corresponds to the face-centered cubic silver and face-centered cubic gold nanoparticles. With these diffraction planes were compared with the JCPDS, corresponding JCPDS. Those who are doing master degree, they can note down this JCPDS. Here in this data bank, we will be having all the crystalline data information, the information about all the materials in the periodic table. So we in which form the crystalline nature of the material is present. So this kind of information we can get it from the JCPDS data bank. So AFM is an important study in the 2D nanomaterial synthesis and the 2D nanomaterial fabrication, because as I mentioned, in this 2D nanomaterial synthesis, the multi-layer material will be converted into a monolayer material. So the multi-layer material will be having high thickness than the monolayer material. So the basic principle of AFM is the AFM will be used to analyze the surface topography of the material. So here we can predict the thickness of the material. So here the monolayer reduced graphite oxide is showing the thickness around 1.2 nanometer, which is very lesser, and which also confirms the formation of monolayer material. The same monolayer reduced graphene oxide, after decorated with silver and gold nanoparticle, here we can see that there is a slight increase in the root mean square as well as the skewness of the material. So as compared to the roughness of the reduced graphene oxide sheets, the roughness of the composites is largely increased. So this roughness will play a huge role in the drug loading. So if the material is having, if a material is having a highly rough surface, you can conjugate or you can load more volume of components like drugs or other molecules. We can load more volume as compared to the transparent or the smooth surface. The increase in the thickness are due to the presence of the metal nanoparticles on the surface of the nanosheets. So here you can clearly, as compared to the other spectroscopic experiments, I think the microscopic examinations will be more interesting and more understandable. And here you can see the transparent graphene oxide, reduced graphene oxide nanosheets that is being formed from the pristine graphite. So here you can see the presence of spherical shaped nanoparticles on the surface of the sheets that here you can see the transparent sheets, and here you can see the sheets that is being uh, incorporated or hybridized with a silver nanoparticle. Similarly, for the gold nanoparticle also, you can see the spherical particles being decorated on the surface of the nanosheets. So another interesting examination microscopic study is the HRTEM. So HRTEM is the ultimate experiment. There you can precisely study the morphology of the nanosheets. Here you can see the transparent nanosheets that are formed uh, by using chrysene as the reducing agent. The same transparent sheets were decorated with a uniform silver nanoparticle. Here you can see the individual nanoparticles were arranged uniformly. I think this slide, I would like to explain the success of polyphenol based nanomaterials. So this kind of morphology you cannot get in the biogenic synthesis, which means the plant mediated synthesis or a microbial mediated synthesis. This is more like a chemical synthesis, that is the nanocomposites that is being synthesized by the sodium borohydride or hydrogen hydrate. These are all strong reducing agents which can be used to synthesize the uniform nanomaterial. But with the help of polyphenol as a reducing and capping agent, we can able to achieve the uniform distribution of the silver nanoparticle. I think the higher magnification images clearly shows the uniform distribution. Even at the corner, at the edges of the nanosheets, the particles are uniformly dispersed, uniformly distributed. So this is the success of our polyphenol-based nanomaterial synthesis. So those who are doing the master students, especially the master students, those who are interested in exploring the nanobiotechnology research and the biogenic preparation of nanomaterials, they can use different other polyphenols for the synthesis of nanoparticle. Similarly, the gold nanoparticle decorated nanosheets also. Here also you can see the individual particles were distributed throughout the sheets of the graphene RGO sheets, and the higher magnification images clearly shows the lattice fringence corresponds to the face-centered cubic gold nanoparticle that is being hybridized on the surface. These lattice fringence can be predicted by the JCPDS value. This is also the lattice fringence, measuring the lattice fringence, as well as the selected area electron trifraction study. These are all the supportive information for your XRD analysis. So another interesting study is the thermogravimetric and differential scanning calorimetry. So this study will give the thermal stability of your nanoparticle or nanomaterial. So here you can see, as compared to the pristine graphite, which is evaporated at around 200 nanometer itself. All the pristine graphite has got evaporated due to the high temperature. Whereas the metal decorated nanomaterials were highly stable and it's sustained around even at 800 degrees Celsius. The material can withstand, which clearly shows the incorporation of the metal nanoparticle on the surface of the nanosheets improves its thermal stability, and moreover, based on the evaporation point of the chrysene, we can able to predict that the chrysene moieties, the presence of chrysene moieties on the surface of the particles and the composites. So after the extensive experiment, the characterization studies, it is very important to analyze their application, to evaluate their application. Because as I mentioned earlier, nanotechnology is a applied science. So all the nanomaterials or nanocomposites that we are preparing has to be applied in different sectors, like right from energy, either from energy application or environmental application, or in the healthcare sector. So here we have evaluated the anti-cancer efficacy of all the nanocomposites, the nano materials, that is silver nanoparticle, gold nanoparticle, and the reduced graphene oxide sheet, and their combinations, the nanocomposite. We have evaluated against two different breast cancer cell lines. And out of all these nanomaterials and nanocomposites, the interesting thing is, all these nanomaterials and nanocomposites have shown increased anti-cancer efficacy as compared to the chrysene. I mentioned that chrysene is a natural anti-cancer flavone. It has been tested with almost all type of cancer cells. Even though the chrysene in combination with the nanomaterial is giving more anti-cancer efficacy than the free chrysene. Among the different nanomaterials and nanocomposites, the silver-based nanocomposites as well as the silver alum nanoparticle is showing better anti-cancer activity as compared to the other nanoparticle. Among the tested nanoparticle, the reduced graphene oxide is having a lesser anti-cancer effect as compared to the other nanoparticles and nanomaterials. So the anti-cancer mechanism was further analyzed with a different staining experiment. Here you can see the AOEB staining, host staining, and the cytomorphological changes of the cells, as well as we have quantified the ROS level ROS level of the cells after exposed to the all nanomaterials and nanocomposites. These studies clearly indicates that the generation of reactive oxygen species by a Fenton-like reaction, it creates oxidative stress-mediated cell death. And I just want to explain the anti-cancer mechanism and the targeting of nanoparticle to cancer cell lines. The targeting of drug to cancer cells is broadly categorized into two types. One is passive targeting, and another one is active targeting. So here in active targeting, it is like a site-specific delivery of your drug, which means it should target only to the cancer cells. So the nano formulation should have a specific ligand or antibody to specifically target a particular antigen on the cancer cells, or a particular receptor of the present on the surface of the cancer cells. So this is a targeted, active targeted mechanism. And another targeting is the passive targeting, So the nanoparticles is randomly targeted. Means because of the the poor lymphatic drainage and the leaky vasculature nature of the nanoparticles, the cancer cells, um, the nanoparticles or the nanomaterial can easily pass through these poor lymphatic drainage, and this process is known as the enhanced permeability retention effect. So because of this effect, the nanoparticles can easily adhere to the cancer cells and release reactive oxygen species. And these reactive oxygen species will hinder the protective antioxidant system, as well as it will create oxidative stress and also the mitochondrial dysfunction and cell cycle arrest. Ultimately, this will lead to the apoptosis, that is the programmed cell death process. So, here I would like to mention that as compared to the cancer cell, the normal cells will be tightly packed, whereas the cancer cells will be having the poor lymphatic drainage. So, which means the nanoparticles cannot pass through the normal cells very easily, but it can pass through easily in the cancer cell microenvironment. So, based on this two mechanism, our synthesized nanoparticles and nanomaterials comes under the passive targeting mechanism. So, apart from the anti-cancer study, we have also assessed the nanomaterials and nanocomposites for its antibacterial property. So here we have noticed that the silver-based nanomaterials is showing better antibacterial activity. And interestingly, we have found that the reduced graphene oxide is also having an improved or an excellent antibacterial property. So we have selected this this particular reduced graphene oxide for the wound disinfection. And we have used Vista rat animal model, and the wound was created, and we have topically applied the reduced graphene oxide for over a period of time, and based on the appearance of the wound, and as well as with the histological studies, we have found that the reduced graphene oxide can be used as a potential wound disinfectant, not only to prevent the microbial infection, as well as for the rapid epithelial regeneration and the collagen deposition, and for the improved skin regeneration. So this material can be further explored in regenerative medicine applications for the preparation of different implants or other tissue engineering scaffolds or something like that. With that graphene oxide, this graphene oxide that is being functionalized with polyphenol can be explored towards regenerative medicine applications. So whatever may be the application, ultimately we have to assess the toxicological implications of these nanomaterials and nanocomposites. Even if you go for cancer therapy, the current existing chemotherapeutic drugs are largely limited due to the adverse side effects. So our nanomaterials also should be checked for their toxicological implications for better therapeutic applications. So we have studied the toxicological implications in Vista rat. We have administered a different dosage of the nanomaterials and nanocomposites, and all the hematological, physiological, and behavioral changes of the animals were studied. And here we have shown the human serum biochemical parameters. Among all the tested nanomaterials, we have found that the silver-based nanoparticles is having some toxic effect on the treated animals, but that is also a meager level of the toxicity. So that is maybe due to the rapid metabolism of the nanoparticles. And another toxicological study is the hemolysis. Here, all the nanomaterials and nanocomposites were tested against the RBC. Based on the RBC lysis, we have found that as compared to the pre-chrysine, all the nanomaterials and nanocomposites were highly hemocompatible. So this study will also convince that the formulated nanomaterials and nanocomposites can be explored for further clinical trials. And this is the histology of all the animals, the animals that is being treated with the different dosage of the nanomaterials and nanocomposites. The histological sections also clearly show no indication of any toxicological effects on their organs. All the organs, right from heart, testes, kidney, thymus, liver, lungs, spleen, heart, and we have also tested quantified the chrysin level in all these organs. So to come to the conclusion, so we have successfully used this chrysin as a potential nanobiotechnologically potential flavone to synthesize different nanomaterials and nanocomposites, especially the metal nanoformulation as well as the 2D nanomaterials and nanocomposites. And this combination will also improve the qualities of chrysin. As I mentioned earlier, the chrysin is having limitations like poor solubility, bioavailability, and efficacy. So the efficacy of this chrysin for anti-cancer and antibacterial applications have been improved a lot by incorporating with it with a nanosystem, and this also opens up several possibilities for several other approach like theranostic device development and other clinical translation. So here I would like to mention that as compared to the free chrysin, the biological activity of the nanomaterial conjugated chrysin is improved. That is which means we have achieved our one of our objective successfully. The similar kind of nanomaterials can be developed for different other nanomaterials, nanocomposites. There are other several myriad of plant polyphenols have been already tested to synthesize different nanomaterials and nanocomposites. The polyphenols as an exfoliating agent, not only as a reducing agent. So from here, I would like to highlight some of the recent breakthroughs in this area of research. So here, in a recent study, the tannic acid, it's a phenolic acid, which is isolated, extracted from the tea plant, and this tannic acid was used as a exfoliating agent. Here also they have used 2D nanomaterial. I think here you can clearly understand the 2D nanomaterials, how it will be converted into single monolayer material. Here you can see the multi-layer material will be converted into single layer material, single monolayer material. Here the molybdenum selenide 2D nanomaterial was successfully sonicated. Here in this study, the role of tannic acid is simply to exfoliate the nanomaterial, not to reduce or not to involve in the reduction process. Here, the same material will be converted into monolayer, but on the surface, the tannic acid will be coated. So by using this tannic acid, we can achieve uh, increased exfoliation rate as well as the stability and the surface charge of the nanoseeds, so that the particles will be stable for over a period of time, and the surface passivation of the polyphenols will be will provide biocompatibility to the nanomaterials. The presence of the biocompounds um, will improve its biocompatibility for various therapeutic applications. And some of the polyphenol based nanomaterials were explored for theranostic nanomedicine. I think for the MSc students, I would like to explain the new concept. I think this will be more interesting for you. Theranostic, the term itself says that the single nano device will be having both therapeutic and diagnostic ingredients. So both therapeutic and diagnostic. So with this combination, a new term was coined, the theranostic. So these theranostic nanomaterials will be having multifunctional multifunction. They are multifunctional, and they'll be having both a diagnostic as well as a therapeutic agent. So these theranostic devices or theranostic nanoformulations can be used for the image-guided therapeutic applications, which means after the administration of the drug, you can monitor the movement of each and every movement of the drug, and whether it has reached the particular target site, or how it is performing. This kind of information you can collect by this kind of a theranostic device, since as we have incorporated an imaging agent to this theranostic device, it will be easy for us to real-time image, do the real-time imaging. So also, for example, if you are using a magnetic nanoparticle that is being conjugated with the anti-cancer drug, with the help of MRI image, you can predict or you can visualize the cancer cell death as well as after the treatment. With an external magnetic field, you can drag the magnetic nanoparticles towards a particular site. There are plenty of theranostic nanomaterials have been developed. The magnetic nanomaterials, that is super magnetic iron oxide, and especially the gold nanoparticle were largely explored for the theranostic in nanomedicine. Here I would like to mention one of the recent research, the polyphenol based theranostic device. Here they have used the mesoporous silicon nanoparticle that is being loaded with gold nanoparticle as well as the doxorubicin as an anti-cancer agent. And after the formulation, the mesoporous silica was surface coated with the gadolinium polyphenol. Here also they have used tannic acid as the polyphenol. The gadolinium polyphenol network was coated on the surface of the mesoporous silica formulation. So once it reaches the cancer microenvironment, due to the acidic pH, the tannic acid will the deprotonation of the tannic acid will release the anti-cancer drug to the cancer microenvironment, and ultimately it will kill the cancer cells. And the presence of the gadolinium will give a magnetic property, so that we can perform MRI imaging, and the presence of gold nanoparticle can be used for the photothermal application, which means the gold nanoparticle is a strong photo absorbent, so it can convert the light energy into heat energy. So that heat will kill the cancer cells. Uh, so here we can use this nanoformulation for both chemotherapeutic as well as phototherapeutic application. On the other hand, this nanoformulation can be used for the magnetic resonance imaging and computerized tomography and photothermal imaging as a diagnostic platform. So there are several metal polyphenol networks have been developed for phototherapeutic application. As I mentioned earlier, phototherapy is categorized into two types. One is photothermal therapy, and another one is photodynamic therapy. In photothermal therapy, we will be using a photoabsorbing agent, either it is inorganic nanomaterial or a polymeric nanomaterial. So this photoabsorbing agent will convert the light energy into heat energy. This is like a sterilization process. We will do bacterial sterilization. We'll apply heat to kill the bacterial cells. The same kind of mechanism will be followed here. But here the nanomaterial will emit the heat. That heat will kill the cancer cells or the bacterial cells. It depends on your application. If you want to develop a photothermal therapeutic device for cancer therapy, so it will kill the cancer cells due to the heat. And if you want to develop antibacterial coatings or hydrogels for phototherapeutic application to kill the bacterial cells, the same heat will kill the bacterial cells and make that environment sterile. Whereas in case of photodynamic therapy, we'll be using photosensitizers. These photosensitizers will absorb the light energy and it will release the toxic reactive oxygen species. That the singlet oxygen will be released from the photosensitizer that will kill the bacterial cells or the cancer cells through creating an oxidative stress. So there is a recent research. In this research, they have used natural polyphenols like tannic acid, catechin, and tannic acid and catechin as an exfoliating agent to synthesize the different transition metal dialkylogenates. These are all the 2D nanomaterials. These 2D nanomaterials were exfoliated, and the polyphenol is being coated on the surface of the 2D nanomaterials. And these nanomaterials were studied for their potential photothermal application towards the biofilm forming bacterial eradication. So here they have developed these surfaces. They have coated these 2D nanomaterials on different surfaces, and they have studied the anti-biofilm eradication property. And this is also the combined therapy. They have conjugated penicillin drug with these nanosheets, and the conjugated nanocomposites can be used for both chemotherapy as well as photothermal therapeutic application. So here, after exposing the nanomaterials to the bacterial cells, they have irradiated the nanomaterial with near infrared light, that is a laser light. So this will convert into the nanomaterials will convert these photons into heat energy, and that will ultimately kill the bacterial cells. So these kind of nanoformulations can be used to develop different medical devices as well as for their surface coating applications. So in regenerative medicine also, the metal polyphenol network can be explored. So in a recent study, the catechin magnesium metal polyphenol network was coated on the surface of the titanium sheets, set titanium surface. This coated titanium surface uh, will give increased improved mineralization and osteogenesis. So we can use this titanium surface for traumatic bone transplantation or traumatic injuries of bone. This can be used as an implant in regenerative medicine applications. So these are all the applications of recent applications of metal polyphenol based surface engineered nanomaterials. There are plenty of nanomaterials were being fabricated, and they have explored for different therapeutic applications. I think this will give some new ideas for the young researchers to explore towards the metal polyphenol. As mentioned by the Richard Feynman, there is plenty of room at the bottom. I say still there is a plenty of room at the bottom. So we have to explore the natural product-based nanomaterials for various therapeutic applications. And I would like to conclude my talk with my recent research. So currently we were developing different types of hydrogels. That is the hydrogels that is being incorporated with metal polyphenol and 2D material polyphenol networks. So these hydrogels are were fabricated towards the antibacterial surface coating as well as for wound healing applications. So this is our current research, and we were working on the hydrogel preparations. And I would like to conclude my talk. So here I would like to thank my PhD supervisor, who is also an alumni of our Kongunad Arts and Science College, Professor Dr. Sivaramakrishnan. So without his constant support, I think most of my research that I have shown was performed under his guidance. So for his guidance and positive approach towards research, I will be eternally grateful. And I would like to thank my current postdoc mentor, Professor Shu, and his research team for their constant support and help. Even during in this pandemic situation, they have helped me a lot. And for the MSc students, I would like to mention that when I was doing MSc in Kongunad Arts and Science College, I'm an average student, so I never thought that I will do this much. I didn't achieve anything, but I will say something that I have achieved. I never thought that at that time, but it is because of my guide, Dr. Sivaramakrishnan, is positive. He has given me some positive energy towards the research, and without his help, nothing would have possible. So I would like to suggest those who are pursuing masters, you can also do better research. I think our UGC also helping you under the leadership of our Honorable Prime Minister Narendra Modi. And now the UGC has framed new regulations, and don't think that if you want to pursue PhD, you have to spend 7 or 8 years or 5 years hardly. And now the situation is completely changed. If you publish 2 international papers, I think within 3 years you can get a PhD degree, and no one can stop you getting a doctoral degree. And I would like to mention that in China, the policy of China also, there for the master students, if they want to graduate, they want to get a graduate in a for master degree, they have to publish two international papers and one native language paper. So think about that. So we have also we should also adapt the same kind of policies, and the master students, you should do some better research and good research, and even before getting master degree graduation, you have to publish your research in a good journals, and that is the success of your master degree, research. And also don't think that yeah, I'm pursuing a master degree in biotechnology, so there is no scope for biotechnology. I think there is not all the jobs are discipline based, and I think no one can stop you because you have pursued your master degree in biotechnology. No one can stop you getting a job or a wonderful career ahead. You'll be having a wonderful career ahead. And see, I did my doctoral degree in biotechnology, but now I'm working as a postdoc in material science department. So science is science. There is no differentiation between biotechnology or chemist or physics. Science is science, and you can perceive good science. And as mentioned by our hero, Dr. APJ, end is not the end. End means effort never dies. So I wish all the MSc students for a better and you know bright future ahead. Thank you.
Thank you very much, sir, for your excellent presentation.
Thank you.
It
Thank you.
was very interesting, and you are
Thank
also sharing your experience with our students. Thank you so much. Sir,
you. Thank
shall
you.
we take a few queries from the chat box?
Sure, sure.
Can I read it out, sir?
Yeah. Please.
Yeah. Question from Meera Darshini. How can the surface properties of nanocarriers can be modified to attain controllable pharmacological and physiochemical features for therapeutic purpose?
As I mentioned in the slides, there are different parameters like the pH, temperature, and their reaction conditions will also play a major role in the tuning of the size, shape, and surface chemistry. And we can we can try different biocompounds for different outcome. With the nanomaterials with different surface functionalized outcome. It depends on your application. So decide the material as well as the reducing agent as well as the capping agent based on your application. So, these are all interrelated. So we can tune coming to the question. We can tune the surface property of the nanoparticle by altering the reaction parameters and also by using different capping agents.
Sir, next question from Karthi Kumar.
Yeah.
What gives best compatibility value of the nanomaterial, charges or size?
Actually, it both charges and size, because charges is largely influencing the stability of the nanoparticle. The size of the nanomaterials will influence on the bioactivity, whereas the biocompatibility is largely depends on the surface property, surface chemistry. If you coat a nanomaterial with a biocompatible polymer, so obviously your material will also have some biocompatibility, certain amount of biocompatibility, and so I don't think the surface charge or the size of the nanoparticles is influenced in biocompatibility. The biocompatibility is largely depends on the surface chemistry, the capping agent or the coating agent that you are using. that plays a major role in the biocompatibility. Moreover, there is a problem with the nanomaterial. You cannot directly administer this nanomaterial, even though it is being functionalized with a biocompound. There are a lot of limitations are there. For example, the protein corona formation. These proteins from the body fluids, this will interact with the nanomaterials, and that will hinder the target specific delivery of the drug. So we have to avoid all these hurdles. For example, we can use zwitterionic or zwitterionic polymers, like the polymers which is having both positive charge and negative charge. These kind of ingredients can be incorporated to get a better nanomaterial with improved biocompatibility and less toxic effects.
Okay, sir. A question from Kanagaraj, sir. Whether is it possible to convert polyphenol oxidase to polyphenol? What are the nanoparticles actively linked with polyphenol?
For the polyphenol synthesis, I have mentioned all the inorganic, most of the inorganic materials, not nanomaterials, can be incorporated with polyphenols. Metal nanoparticle, the reaction and other things, it depends on the material that you are choosing. If you are choosing metal nanoparticles, you have to go for the reduction reaction, or if you want to explore for the 2D nanomaterials, you can simply use these polyphenols as a surface capping agent or the exfoliating agent in order to increase the exfoliation rate. So it depends on the material that you are choosing. You can functionalize through the covalent bonding, or simply you can coat the the polyphenols in a non-covalent bond formation on the surface of the nanoparticles. You can explore all the inorganic nanomaterials or materials. In the periodic table, you can see recently people are exploring the black phosphorus, that is miraculous, that is considered as a miraculous nanomaterial. It comes under 2D nanomaterial. So this kind of nanomaterials, new currently the materials that is in trend, you can explore these nanomaterials for your applications.
So, question from Karthik Kumar. What is the role of temperature in green synthesized nanoparticle during synthesis process? And is there any possibility to increase the zeta value of the material to use polymer doping agents?
Yes, obviously, if you use the cationic polymers or anionic polymers, you can achieve more zeta, means surface charge of the particle. So you can use these polymers as a doping agent, and as well as the other question is, what is the other question? Hello?
Yes. And is there any possibility to increase the zeta value of the material?
No, I have answered for that question. The first question.
Role of temperature in
the temperature will play a major role. So it will increase the solubility of the metal precursor, as well as the reaction rate of the reducing agent. And the nucleation growth of the nanomaterials will be largely influenced by the temperature. So we cannot say the increasing temperature will always give increased yield of the nanoparticle. So at certain point, the particle will get decomposed at a certain temperature. So we have to be very much careful about the fixing temperature.
Question from Shrikant.
Yes.
Microsynthesis of silver nanoparticles in liquid medium for XRD characterization, they want the materials in powder or crystal form. Can we convert the liquid material to powder or crystal samples for XRD characterization?
Powder form is most preferable, and you can prepare your sample using different surfaces, either a titanium surface you can use or a glass surface. You can coat your powder on the on that surface, and then you can perform the XRD analysis. My suggestion is powder form is more convenient and more preferable. Even in any synthesis, whether it is microsynthesis or phytosynthesis or any other synthesis, chemical synthesis, powder form of the sample is more preferable for XRD analysis.
A question from Suganya. How is the microorganisms used in the biological synthesis of nanomaterials?
So again, the microorganism also will synthesize certain metabolites like proteins and the reducing sugars. These metabolites will involve in the reduction and surface capping of the nanomaterials. In that way, microbial synthesis. But the problem, as I mentioned in my slide, the problem with the microbial synthesis is you have to go for a multiple purification step and tedious downstream processing, and the purification is you have to there will be impurities in the nanomaterials as well, and we know that maintaining the microbial culture without any contamination and these things is a very tedious process. But as microbial mediator synthesis, the proteins and other reducing sugars and the metabolites that are being synthesized by the microbial cells are largely involved in the reduction and stabilization process.
Okay, a question from Janani Shri. Sir, what is the half-life of polyphenols in drug delivery?
This is a very interesting question. So far, we have not checked the half-life of the drug, but based on the dosage of both the polyphenol as well as the polyphenol that is being conjugated with the nanomaterial, we can minimize the dosage and we can avoid the adverse toxic effects. And we have not checked the half-life because the problem with our studies, we did a surface functionalization. This kind of half-life studies can be performed only in if the drug is being encapsulated with the polymer nanomaterial. You can check the drug release kinetics and drug loading content, all these parameters in case of the polymer-based encapsulation formulations. But here we have used the surface functionalized nanomaterials. I mentioned that the polyphenols, even after it is being functionalized on the nanomaterials, the structure of the polyphenol itself is converted into quinone form. So we cannot trace the amount of the polyphenols, or we cannot check the stability or the half-life of the nanomaterials. Maybe in future, these kind of experiments can be achieved.
Question from Akshaya Krishnan. Does nanoparticles of silver will be immunogenic in nature, and will it act as biomarker?
Yes, we can use this silver nanoparticle as biomarkers, but directly we cannot use. But this silver nanoparticle is having an interesting property called surface enhanced Raman scattering. This particular property can be used for the imaging of a particular cells, either it is a cancer cell or a bacterial cell. We can use to image for imaging and as well as for the diagnostic applications. We can use silver nanoparticles alone. But in case of biomarkers, we have to use some analyte with this. We have to conjugate or we have to functionalize any the analyte with this silver nanoparticle, then only we can make a better biomarker.
Question from Akshaya Krishnan. By doing photodynamic therapy, does it lead to production of free radicals?
Yes, obviously, the basic principle of photodynamic therapy relies on the generation of the reactive oxygen species, the singlet oxygen, that will create oxidative stress mediated cell death. Either it is a bacterial cell or cancer cell. The photosensitizers, when you irradiate the photosensitizer with a particular wavelength of laser light, these photosensitizers will release toxic reactive oxygen species, that is the singlet oxygen, that will ultimately kill the cancer cells or bacterial cells.
Okay. How can we get uniform size and shape through green synthesis process? Question from Karthik Kumar.
Yeah, I mentioned in my slides, you have to optimize all the reaction conditions like pH and mainly the stoichiometric proportion of the both precursor as well as the reducing agent in what combination and how much reducing agent you are taking and how much how much quantity of the precursor that you are taking. These parameters will largely influence the size shape of the nanoparticles. We have to fix, even if you synthesis the nanoparticle using a plant extract or a microbial extract, we have to fix all these parameters. So these parameters will largely influence the size and shape.
Okay. Kanagaraj sir has asked, does the polyphenol-based nanoparticles have antiviral property? If yes, did you check it for COVID-19? SARS COVID-19.
Yeah, it's a very interesting question. Yeah, we can use this polyphenol metal network to conjugate with a different. We know the World Health Organization and our ICMR have declared to use the chloroquine and remdesivir as for the COVID treatment. So we can conjugate these two drugs. The main problem with the hydroxychloroquine is it is having adverse side effects and the cardiac arrest and other sort of adverse effects it is showing. So they are limiting the usage of this particular drug. So we can incorporate these drugs with the metal polyphenol networks. We can conjugate these drugs and we can reduce the dosage of these drugs, and we can avoid the adverse side effects, and we can develop a better treatment for the COVID-19.
Okay. In FTR characterization, we can predict all the functional groups of solutions and extracts, but when it is converted into a synthesized metal oxide particle, some of the groups are difficult to find compared with the raw material. So,
which means maybe if the polyphenol or the biocompound or the reducing agent or the capping agent, if it is there on the surface of your nanomaterial, obviously you will get the functional group, the corresponding functional group. If you cannot predict in the FTR analysis, better you can go for the X-ray photoelectron spectroscopy, which will give a clear outcome about the complete composition of your nanomaterial and nanocomposite. So that will be useful in your case. You can perform X-ray photoelectron spectroscopy.
okay. Dr. Bhuneshwari ma'am, is there any other questions in the chat box left?
Maybe few questions in the beginning you can see. Like Vishnu.
Yes, please. Yes, please. Go ahead.
Dr.
How to predict the size of the nanoparticle from combining the results of FESIM and XRD characterization?
You can use, sorry for not mentioning that, we can use Image J software for to measure the size of the nanoparticles, either it is from FESM or HRTM. You can use the Image software, and uh, you can also find videos in YouTube that how to use this image say, software to measure. These are all the simple steps. Only 3-4 steps are there. It is not a very tough thing you can do by yourself. The main thing is you have to get the software, image software. If you have that software, and you can easily measure the size of the nanoparticle. And if you take one micrograph, uh, you just measure all the nanomaterials and all the nanoparticles, and you just measure around 50 to 100 nanoparticles in that particular micrograph, or all the micrograph that you have taken. So you can get the average size in the same software itself. There are some options to get the average size as well as the mean size as well as the standard deviation of that particular distribution. So all this statistical analysis can also be performed with the image software. So if uh, if you are doing that FSM or HRTM in that instrument itself, they will be having these software, but that will not be more convenient for you at the time of experimentation. So you can use the image software leisurely, and you can measure the particle size, and you can plot the graph in the hydro. You can find the particle size distribution, and you can plot the histogram. Note it down, image.
Okay,
The
thank you
software
so
name is
much.
image.
Okay, thank you so much for clarifying all the queries.
Thank you.
You.
Thank
It
you.
was
It
a
is a
very
pleasure.
nice session.
Thank you.
And now I request our ever supporting Dean Academics, Dr. S. Madan Shankar sir, and one of the prestigious alumnus of our esteemed institution, to share few words. Sir, please.
Uh, Good evening.
Good evening, sir.
Good evening. Good
Good evening.
evening. Good evening, Dr. Amsaveni, Dr. Bhuvneshwari, and I think Dr. Satish Kumar is our alumni, right?
Yes, yes, sir.
Which batch?
Uh, 8 to 10, 2008 to 10
8
master.
to 10.
MS,
Yes.
Okay.
MSc
PG.
Biotechnology.
Okay. Okay.
Yeah.
Okay. So I've been listening to your talk right from say about an hour and a half since.
Yes.
It's a very wonderful lecture that you have given. I'm so
Thank you.
happy. I'm so happy that you have landed at a nice place and you have done good quality of work.
Yes.
And not only that, you have taken pains to share every bit of information to the participants. It's
Yes.
really awesome and it's massive.
Thank
I
you.
congratulate you for giving a very vivid presentation on nanotechnology. I remember all this when I framed the syllabus at PSG Arts in 2010. 2010. Even then, these terms like theranostics and all this, I learned and I taught my postgraduate students in 2010. So I'm happy that you have worked on all those aspects with a lot of hands-on experience. At this point of time, I only remember one of the great writers, Robin Sharma. He always in his books are very unique, and he says the quality of success you will experience in your life ultimately depends on the tiny choices you make every minute, every hour of every day. And I'm sure that Dr. Satish Kumar has taken a very right choice. Firstly, for joining Konganadu Arts and Science College in the Department of Biotech. And I'm sure that this learning that you had in 2 years from this prestigious institution would have given you a lot of inputs to set a right stage for your research and further down the line. I'm also thankful and happy that Dr. Amsaveni has taken a lot of pain in arranging this lecture in a very short note. And I could see somewhere about 170 plus participants who have been benefited by the lecture today. And as an alumni of this department, as an alumni of this institution, I only wish that more and more people, alumni, contribute in knowledge sharing to the upcoming generations, and this will add value and flavor to their research aspects, and it will also give them an impetus into how they can go into a focused research, and also what kind of path they can traverse to reach successful heights. And I'm sure that the department, with all these alumni as a support, will scale peak to peak in coming years, and we would be one of the excellent department. And my aim is that the department should be an industry-sponsored department in about 5 to 10 years from now. So I request all our alumni to support us in this endeavor, and personally congratulating Dr. Bhuneshwari, Dr. Amsaveni, and the team of faculty from the Department of Biotech, who have been very supportive and very nice in organizing all these programs. And my special thanks and appreciations to the chief guest of today, Dr. Satish Kumar, for giving a very, very vivid presentation about the application part of nanotechnology, and uh, the very fine details about. I'm very happy that there was a lot of questions from the participants, and you have taken pains to answer every bit of questions that was asked in a very nice way. Thank you so much, and wish you good luck and have a nice evening.
Thank you, sir. Thank you for the kind words. I'm really overwhelmed by your words. Thank you. And at this juncture, I would like to thank Dr. Ramsavini and Dr. Bhuneshwari for not, I won't say for inviting me, because as an alumni of Kongunad Arts and Science College, it is my
This is our home.
pleasure and privilege to be back at KSC to share my research experience that I have gained over a span of 8 years. So again, I would like to thank all the organizing members, committee members, and our respected Dr. Arichan sir for giving me this wonderful opportunity. Thank you. Thank you. Thank you all for listening to my talk.
Okay. Thank you, Dr. Madan Shankar sir, for your valuable presence and gracing this occasion. Now I request Dr. V. Bhuneshwari ma'am, Associate Professor and Head, UG Department of Biotechnology, to honor our resource person with an E-certificate. Ma'am, please.
Sir, thank you, sir, for providing an interesting and wonderful presentation.
Thank
And
you.
it's a really very informative session. I would like to highlight some of your words. You have highlighted about the nano in medicine and synthesis of nanoparticles using different methods, especially about biological method using plant extracts and polyphenols, particularly chrysin. And you have interestingly said about this density functional theory done by your collaborator Dr. Satish Kumar sir. And you have also highlighted about the 2D nanomaterials for skin regeneration. And you have said about this gold nanoparticles for photothermal applications and in cancer therapy, and and act as a photo observing agents and emission of heat to kill the cancers, and even the nanoparticle applications in MRI and CT scan. Thank you very much, sir. And I hope you will accept our collaboration with our college, as most of us are working in this area. And thank you very much, sir. On behalf of Kongunad Arts and Science College, we are very much delighted to honor our chief guest with E-certificate as a token of love and gratitude. Sir, please.
Thank you, ma'am. Thank you. And those who are pursuing MSc, the master for their master dissertation, if they want to, do research in nanotechnology, they can approach me. And also, I would like to have a collaboration with the faculty members of our department. So in future, surely we'll be having a collaborative. We can have a collaborative research.
Thank you very much, sir.
Thank you.
The same will be emailed to you, sir. We are
Thank you,
offering
ma'am.
this.
Thank you.
Okay,
Thank you.
thank you.
Very kind of you. Thank you.
Thank you, Dr. Bhuneshwari ma'am. Now I request Dr. Gogul Ramnath sir, Assistant Professor in Biotechnology, for the vote of thanks. Sir, please.
Am I audible? Hello. Good afternoon. Good afternoon, all. Hello.
Yes, sir.
Am I audible? Good evening, all. Myself, Dr. M. Gokul Ramnath, working as Assistant Professor in Department of Biotech, Konad Arts and Science College. On behalf of PG and Research Department of Biotechnology, it's my great privilege to deliver a vote of thanks in this occasion. My heartfelt thanks to our President, Dr. M. Archami, sir, who has been the inspiration with his dedication for the progress of our institution. I sincerely thank our beloved Secretary and Director, Dr. C. A. Vasigi ma'am, who has given her immense support and encouragement in all our endeavors. I would like to thank our respected Principal, Dr. M. Lakshmanaswamy, sir, for his valuable support and guidance. I express my gratitude to our dynamic person, Chief Executive Officer, Dr. V. Chinnaswamy, sir, for inaugurating this webinar with his valuable presence. I also thank our Dean R&D, Dr. S. Paulswamy, sir, for his constant motivation in all our activities. I thank our kind and courteous person, Dr. S. R. Madan Shankar, sir, Dean Academics, come Head in PG Department of Biotechnology, with his valuable presence and nice words. I am so delighted in thanking our today's research person, Dr. G SATHISH Kumar, who spared time from his busy schedule and enlightened us with his excellent presentation. I also extend my thanks to the organizing secretaries, Dr. V. Bhuneshwari Madam, Associate Professor cum Head in UG Department of Biotechnology, and Dr. R. Amsaveni Madam, Assistant Professor in Biotechnology, for organizing such an excellent webinar session on this emerging field. I also thank all faculty members of our department for their kind and constant support. I finally thank all the participants for their cordial cooperation. I hope you all will give the same support for all our upcoming webinar sessions. Thank you once again. Thank you all.
Thank you, Dr. Gogul Ramnath sir. I thank all the participants for your active participation. Special thanks to Dr. Satish Kumar sir.
Thank you. Thank you, Amsavani.
Thank
Dr.
you.
Amsavani. Thank you.
Thank you, sir. Thank you, sir. Dr. Bhuneshwari ma'am, shall we conclude, ma'am?
Yes, ma'am. We can.
Yes, ma'am. Thank you all.